Hey, welcome to Buy 100 Day 2. We're going to do some work on cells today, and before we get into that, I just wanted to address some of the concerns that had come through on Learning Suite from you. So, in Learning Suite, we've had some issues that are actually beyond my control. So, this had nothing to do with me or anything I'm doing or the course. It's actually just a problem I've had across both my courses with Learning Suite, where it was published and unpublished and published and unpublished throughout the day. So some students were able to get content, some students weren't. Some students were able to get it in the morning and not in the afternoon, but then again in the evening. So I really apologize. I think that's been sorted out. Let me know if it hasn't. I'll try and get to the bottom of that. Other students have had questions about what kind of schedule should they follow. So in the course documents, I've put in a suggested schedule, and that's generally pretty close to the schedule that we follow when we have the class in person, and that is we cover about a chapter or two a week. So go in and take a look at that. I've taken off any due dates or things like that, but just wanted you to kind of have this idea of what you might want to cover in week 1, week 2, week 3, just so you can keep up with the course if that's helpful for you. But in all reality and in all honesty, you're welcome to just take whatever pace you want in this course. You could work really hard in the first several weeks and knock out a lot of this course. You could wait till the end and knock out a lot of this course, which is probably what a lot of you will do, which is totally fine. But I would suggest that you just methodically go through at a set pace to finish the course, so that you don't have to think back. Okay, where was I in September? Now that it's December 1st and I have to wrap this class up in 10 days. The one thing that won't be coming regularly, so all the courses there, we're currently putting in the midterm exams that you'll take via Proctorio. So that will all be in there hopefully within the next week to 10 days. But the one thing that won't be there super fast are the recorded lectures, which I'm trying to do about one a day, but some days I won't be able to. And so the goal is to have these done by the end of September, so that you can go in and truly take this course on demand and get whatever out of it you can with the lectures. Some of you can see as you kind of thumb through the lectures that I've structured them for this semester. I've structured them in a way that you can actually get quite a bit out of the lectures, if not everything out of the lectures, just by reading through them, carefully considering them, pausing, taking time to think about what's being said. That will really help you when it comes to the exam time. So the recorded lectures aren't absolutely a necessity, but you'll definitely get pointers and bits and pieces that would be helpful in terms of even just remembering topics by the recorded lectures. But some of you have a deep background in biology, and you may not need to wait for the recorded lectures, and that's totally fine. So in terms of quizzes, there are 3. So there's what we call the chapter quiz, which is a quiz over the contents of the chapter. And it should be pretty straightforward, pretty easily done. It's open book, it's open note, it's open neighbor, right? If you want to make contact with somebody in the class and work through these quizzes together, even in groups. The one thing I will say about the chapter quiz is make sure that it's not something where, hey, neighbor, will you take the quiz this week and then pass me the answers? I'll take it next week and pass you the answers. That's not what we're after. We've had that problem in the past. And unfortunately, students who take that approach usually don't perform super well in the class, and it is a tiny bit dishonest, let's be honest, right? So let's not game the system that way. Please do take the time, even if you're working in groups, for all of you to look it up, get the answer, and mark it down. And then we have what we call a quiz for the reading, and this is just whether you go in and you report how much you read. I read 25, 50, 75, 100% of the chapter. Put it in there. We just want you to get points. So even if you bomb the chapter quiz, we still want you to get credit for having read whatever portion of the chapter that you did. And then this last one is called watching. So there's a quiz where you can go in and say, hey, I watched 100% of the lecture, 75, 50, 25, whatever. And you'll just get points for having watched the lecture. So we're just trying to build points in that you'll get automatically just by putting effort into the course. Now, with the watching, the one thing I will say there is because the lectures aren't already on demand and they'll be coming out throughout the semester, if you would like to go through the lectures on your own, as long as you go through the lectures on your own, I'm totally fine if you want to just go in and give yourself credit for having watched the lectures. In my opinion, the way I've structured the lectures, it's very similar to watching, although as I mentioned, for a lot of us, watching the lectures might be the best way to get information that we want and help put things in our long-term memory. So if there's any questions, go ahead, head over to Learning Suite and just put those, send them as an email to me through Learning Suite and with your TAs, and your TAs and I will jump in and respond to those. So I'm going to go ahead and close out this video here, and we'll just get into the lecture. So there's this link that I saw several years ago, and we have to ask ourselves, what does this have to do with biology? So we're going to wait for this to load. Tried to load it before class, but here we go. Don't worry, this is just an ad, and I apologize. Hopefully I can edit this out later. If not, you just got a nice IBM
All right,
commercial.
so it wasn't even 2 decades ago. Astronomers knew of no other solar system than our own, right? Well, astronomers are now saying they believe 100 billion, that's right, billion stars in the Milky Way, all have at least one companion planet. All right, Chad, that's pretty cool, because you and I, right, we learned 9 planets.
Yes.
We grew up
Right.
learning the 9 planets. How did this happen? How did we...
And remember, there was one past Pluto, but they didn't know. Then we lost Pluto. It became a non-planet. It's like, oh my. Now we're to 100 billion. We found this out. These numbers are now coming from scientists saying, look, we have Kepler. Kepler is looking out there, and Kepler is finding at least one planet per star. Every star that it looks at, it's finding at least a planet around that. And then there's this micro-lensing system out there that's finding more stars beyond stars in our Milky Way galaxy. So adding up to about 100 billion stars in the Milky Way, each having at least one planet. That's easy multiplication. That's at least 100 billion planets out there.
So I got to ask you this, though. Does this mean that there's the possibility, life, companion life,
It's
other life
past that.
beyond the
I think
universe,
it's
not just us?
probability. Certainly. All you really need is water, something warm enough, something in that at 60, 70, 80 degree range here, Fahrenheit, and you have a planet with some type of life on it.
I like that idea.
There's a lot of chances of that.
I like that.
Yes.
I've always believed that could be possible,
Sure.
that we weren't alone.
Yeah.
All right, thanks, Chad.
You're welcome.
So what I want you to do is just take a second and ask yourself, what does this have to do, what does that clip have to do with biology? And just go ahead and hit the pause, give yourself 10, 15 seconds to think that through. What does this clip have to do with biology? So hopefully you're back now, and one of the things that you can think about with this is, what does life require? So we know that throughout the universe, with all these stars, that there's at least one planet per star, which is fascinating to think about. And as they mentioned in the clip, right, the probability of life is almost a certainty, and that we're past that now. And the question is, how do we find it? How do we get there? Which is really interesting. But think about, what does life require? Oh, and just as a side note, I should say, I think it's really cool that science is finally to the point where our own church doctrine was, 200 years ago or 150 years ago, where we talked about how many different planets there were. It's pretty cool. Pretty cool to see that our doctrine embraces science. So the question, what does life require? What are the characteristics of life? I want you to think about those just for about 15 seconds and try and work those through in your mind. So some of the characteristics, we'll come to what does life require in just a minute, but some of the characteristics of life are something like this: growth, metabolism, and homeostasis. And we'll cover these throughout the lectures in this course. But talking about specifically what does life require, well, we all know that it requires water. We can't go long without water. So water is this really amazing molecule that has 2 hydrogens and 1 oxygen atom. your body's about 65% oxygen as a result of this. So water is a solvent, right? And you have these two different options, right? A solute and a solution. What is being dissolved is the solute, and the solution is the solute in the solvent, so salt water, right? So if you put salt in water, the solute is salt, and the solution becomes salt water. Makes sense, right? Pretty simple. Water is what makes things go. We have chemical reactions with hydrophilic molecules, those are water loving, and the hydrophobic, which are water hating. Water forms hydrogen bonds. So these are just really weak attractions that happen between the molecules, and this is what makes water so easy to ebb and flow. Water tends to stick together. This is why you can put a droplet on the table and you get a little bubble, right? You almost get like this quote-unquote skin, and that's because water is lightly cohesive. And one thing I would argue is just that water is perhaps the most unifying, important, and powerful molecule on Earth. Just saying, right? I mean, all you have to do is look around where we live in Utah, and you can see the effects of water in terms of erosion, lakes, mountains, streams, all that kind of stuff. And you think about the Grand Canyon largely formed through water. Think about a lot of the major structures that we see on Earth, and water had a huge role. The other molecule that we'll talk about is carbon. So carbon is the basis of all life on Earth. We're all carbon-based life forms. Carbon is the basis of life because it's almost like this molecular tinker toy, which basically means that it can bond 4 different atoms at once. About 18% of your body mass is carbon. It forms very strong bonds as opposed to water, which are very weak. And these bonds are covalent bonds because they share electrons. And carbon, most importantly, carbon can make macromolecules, which are these large stringed molecules. And can you think of any examples of macromolecules off the top of your head, maybe from your reading? So the first one that we'll hit on are carbohydrates. You guys have heard about these all the time. You almost always hear they're bad, right? Carbs are bad, eat less, eat less. Well, carbs are what run your body, actually. And if you cut carbs out, your body will run off of fat, which is some of the more popular diets these days, where you reduce carbs to the point that your body runs off of fat. But in reality, carbohydrates are central to our body's functions. They are, of course, carbon-based, but they have oxygen and hydrogen as well. And you can see they can form these really long chains and sugars, essentially, that are broken down. We'll talk more about that later on. It's our major source of energy. It's a set structure. We often get them out of plants, and starches is our major source of carbs. So protein. This is the stuff you hear, hey, you need more protein all the time. You got to have protein, right? So also carbon, hydrogen, oxygen-based, and nitrogen. And these are just long strings of carbon-based molecules that fit together to form proteins. And these molecules that are fitting together are called amino acids. They're joined by peptide bonds, which are another strong bond. There's about 20 amino acids in total, and different combinations are going to have different functions. Another macromolecule are lipids. These are the fats, basically. They're composed mostly of carbon and hydrogen. They're hydrophobic, right, because they're fat. So you think about putting olive oil in water, olive oil and vinegar, they separate, and that's due to the hydrophobic nature of oil being fat. So you have 3 types. I wouldn't worry too much about the 3 types, just kind of showing you that lipids come in many different types. And we have the macromolecules, the nucleic acids. So these are chains of nucleotides. You should be pretty familiar with these. You're composed of them. So we have both RNA and DNA. Nucleotides have a sugar, a phosphate, and a nitrogenous base attached to them. And they come together to form really long strings of these nucleic acids, both RNA and DNA, although RNAs are generally much shorter. And there are some rules to how these molecules come together. So especially for DNA, DNA is double-stranded, and the bases come together in a specific way. So A pairs with T and G pairs with C. Never will G pair with T or C with A or any of those combinations. It's always A with T and G with C. Some of these molecules have structure. So, for example, with DNA is a double helix, and it wraps around each other as it's made, and forms almost this cool stairwell structure where you've got these backbones made out of phosphates, and then these rungs are made out of the nitrogenous bases, and these are going to be your A's, T's, C's, and G's. So let's take a second, let's do a little activity. If 60% of your body is oxygen, right, and we've said 60-65% of your body is oxygen, 18% carbon, 20% hydrogen, and 2% nitrogen, how much of each in pounds does your body have? If you're a little bit self-conscious about your weight, that's totally fine. Just do it for a 9-pound baby, and let's figure out what composition of that individual is these different elements. We're not going to work that out together, but hopefully you'll do it and see that it actually adds up to quite a bit of weight that your body's composed of. Let's also move on. We're going to move on to some of the structures in the cell, but first, before we get in, I want you to kind of lay the foundation for yourself. Hopefully you've done the reading, but what we're going to do is we're going to have you pick an organelle. When we're in a class setting, we'll have everyone pick an organelle. We'll divide the class up into organelles, and then they'll go in and they'll draw it, and then they'll find the function. But what I want you to do is take this sheet right here, and you're going to go in and just write down. So you'll come in and you'll say, okay, plasma membrane, this is its function. It's found in animals, plants, and bacteria, or not, right? Is it only found in animals, only plants or bacteria? Is it just in animals and plants? And I want you to kind of pull this together so that you have this to study from as an example going forward and in preparation for the exam. So you should have this piece of paper. It's on Learning Suite right underneath this lecture. There's a backside to it that has more organelles on it, and it also has 3 circles, and those circles will help you kind of take the information from where it's contained here, and you can write whether it's present in all 3, whether the organelle is present in all 3, or whether it's present in 2 or just 1, using the circle diagram, and I'll show you that right at the end of this lecture as well. So why don't you take about 20 minutes and just go ahead and do all the searching in your book, on the internet. If you're working with a neighbor, that's also fine. Figure out kind of the function of each one of these organelles and where it's found. Okay, I'm going to keep on. So here's kind of what we're looking at. Here's some of the functions, some of the organelles that you should be able to see. And what we're going to do now is I want you to take that cheat sheet that you just created from the class, and I want you to go through and figure out which organelle is most closely related to a public library, which one's most closely related to the U.S. Postal Service, a water tower, coal burning, power plant, right? You kind of get this idea. I want you to match up organelles with these ideas. And I know this kind of seems really silly, but you should take a second here and really invest in this because it'll help you remember later on. So go ahead and hit pause and work through those. Okay, hopefully you're back. Here's what we have. So the library, right? That would kind of be like the nucleus. It stores information in DNA. The U.S. Postal Service, what do you guys think? Golgi apparatus, it kind of sorts and delivers material. A water tower would be a vacuole. Coal burning power plants is pretty easy. Mitochondrion, right? Converts fuel to energy. And then we have the solar power plant. So it also should be pretty straightforward, right? It even has the word plant in it, right? So that would be the chloroplast. And then manufacturing plant would be the ribosome and ER. These build proteins. Now there can be a little bit of wiggle room here in your responses, but as long as you can justify your response with what you've found and know to be true, we're cool with that. And a moving company, something like the centriole, right? These are going to move chromosomes during cellular replication, so we would call them our moving company. Okay, so let's talk about the major types of cells. So you have the prokaryotic cells here, which are not so attractive, often very tiny, and eukaryotic cells, which can also be unattractive and tiny, but also add up to something incredibly adorable, right? So talking about the two different types, all cells on Earth are either prokaryotic or eukaryotic. So karyos means kernel, that there's a nucleus. So you kind of have this pro or before the nucleus, and you have the U or the true nucleus, and that's the difference between these two cells. It's not that one's single-celled and one isn't. You can also have single-celled eukaryotes, but those eukaryotes will have a really well-formed nucleus. So some of the prokaryotic cells here are just very basic, right? You can kind of look at this figure, how they're set up. They're small and simple in structure. They don't have a true nucleus. An example is bacteria. Eukaryotic cells, as we mentioned, are much more complex. They have the nucleus, and as I mentioned before, right, they can be single or multi-celled. I already mentioned they have a nucleus, and then they have the organelles with specialized jobs. Now, sometimes prokaryotic cells can have some element of organellular work for sure, and you'll see that as you do the cheat sheet. But what's different between them and eukaryotic cells is that there are more organelles and they are highly specialized in their jobs. So don't be thrown off again. Remember, things like yeast are eukaryotes, amoebas are eukaryotes, all the way up to really complex plant and animals. All cells have a plasma membrane, which is this element here, right, where you've got the hydrophilic heads on the outside and the hydrophobic fatty acids on the inside, and that causes them to essentially create this phospholipid bilayer, where these portions in here, the hydrophobic fatty acids, are hiding from water essentially, and the hydrophilic portions are turned in towards the cell and towards the outside environment. And this forms what we call a fluid mosaic, which means that these pieces are not stuck together. They can actually rotate, and things that cross this layer can move around between it. And it is semi-permeable as well, which allows things in and out. And there's a YouTube link that you can follow here and watch as well. And we'll try and watch this one right now.
When comparing between prokaryotic and eukaryotic cells, let us first recall that eukaryotic cells include both plant and animal cells. So, the first comparison is that the DNA is found loose in the cytoplasm in prokaryotic cells, whereas in eukaryotic cells, the DNA is contained within a nuclear membrane, collectively forming the nucleus. Now, the second point is that within that nucleus, the DNA is wound around histone proteins in a eukaryotic cell, but in prokaryotic cell, the DNA is naked. It is not wound around histone proteins. The next point is about the mitochondria. The mitochondria are represented here in eukaryotic cells. These are the site of respiration, but they are not contained in prokaryotic cells. All of these little dots that you see around here, these all represent ribosomes. They're present in both eukaryotic and prokaryotic cells, but what the IB really wants you to know is that in a prokaryotic cell, the ribosomes are 70S, and in the eukaryotic cells, the ribosomes are 80S. Basically means they're a little bit bigger, but the IB actually wants you to know those numbers. Finally, we've already pointed out the nucleus here surrounded by a nuclear membrane. That makes it a membrane-bound organelle. Also, the mitochondria are membrane-bound organelles, but to include in that, you have the rough endoplasmic reticulum, the smooth endoplasmic reticulum, the Golgi body, or sometimes known as the Golgi apparatus, and also the chloroplast in the plant cell. These are all membrane-bound organelles, but a prokaryotic cell does not contain membrane-bound organelles. These little things here are actually plasmids. They are not membrane-bound organelles. Prokaryotes do not contain membrane-bound organelles.
So hopefully that was helpful, and should build on what we talked about already. So back to the cell here. we've looked at this several times now. I would hope that you'd spend a little bit of time here and not worry so much about memorizing this entire thing. You certainly do need to know the names of the different organelles, but just kind of thinking of a way, like, what's the function of these organelles? What are they doing? And certainly memorization will play a part in that. But if you can understand and liken them to something, right, like the nucleus is the library, right, it'll be much more helpful for you when you go to apply your knowledge later. And let's do some of that right now. So imagine there's a 5-month baby boy, comes to the ER with chronic diarrhea and symptoms of malnutrition. The mother says she has been feeding the child regularly, but he is much smaller than he should be. An examination of his stool sample shows that they are foul-smelling and greasy in appearance. You decide to perform a blood test and find that he has neutropenia. Oof, scary. I should know that. I've read that like 500 times by now. Anyway, this comes out to a low count of white blood cells called neutrophils. You biopsy the baby's pancreas and find low levels of digestive enzymes and other cellular proteins in the cells. What organelle dysfunction is most likely to lead to these symptoms and why? And I want you to just think about that for a minute, okay? Hit the pause button, look at your cheat sheet, really work this out. I'm not going to give you the answer right now, and I want you to be able to kind of think that on your own. And the point here is there is an organelle that we have in mind, but the point is to look at your organelles and justify which one could be causing this issue. So go ahead and hit pause, think it through. Okay, the second one here is a 2-year-old girl that comes to the ER with coughing, recurrent episodes of pneumonia with increased sputum, right? This is mucus. She also exhibits an inability to gain weight and is much smaller than her peers. Her mother tells you that the child has complained of her skin tasting salty. You perform a test that reveals excess chloride leaking outside of cells. So what organelle is not functional here, and why? And as you work on these things, I want you to think about not only organelle but why. You really have to justify what's happening here. So you're applying. It's not just memorization. You're applying what you know. So go ahead and hit the pause button and work through this one. Okay, so I mentioned to you earlier, and if you've printed off the handout, you've seen that on the back of the handout, you have these 3 circles, right? When I've given examples where we have our prokaryotes, or maybe like a bacterium is what you can think about, and then some plants and animals. And what you want to do is you want to put the organelles that exist here between all 3 in the middle. Maybe there's an organelle that's just between plants and bacteria, another one that's between bacteria or prokaryotes and animals, and others that plants and animals share. So just put all your organelles here, and you'll really be able to understand who has what and who doesn't, and who shares what and who doesn't. And that'll be it for today. So hopefully you got something out of this lecture. Let me know if you have any questions. Put them on Learning Suite. I'm happy to respond. Have a good one.